So, I've never given a public lecture about this topic before. I think I need to lower this a little. And if I went to a talk with this kind of title, then I kind of would expect that I would hear something about how physics, a powerful science, is going to help us cure AIDS. You might know somebody who has AIDS or who is HIV positive. A reasonable thing. You're going to hear nothing of the kind. So I apologize for those of you, and you would be entirely justified in walking off if that was your expectation, but I hope you'll hear me out. I need to make a second disclaimer. The other people in the audience are physicists, okay? And you're going to not hear anything about string theory, field theory, or even quantum mechanics. You're not going to hear about statistical physics, all the things which justify this wonderful place. So I too hope you'll hear me out. So this type of work really originates in the life sciences. After the AIDS disease appeared in the later part of the 20th century, there has been an enormous amount of research done in the life sciences about the HIV virus. So we know more about the HIV virus than just about any other viruses, by an enormous amount. The methods which are being used are ever more sophisticated, but there are many unresolved problems. In fact, the more we learn about HIV, the less we begin to understand it. It's like a maze. There is a perspective, and then you think you understood it, and you see a new perspective. So HIV also gives us a way of understanding things which we didn't do before because we've studied it some more. And one of the big payoffs is that we now are using the HIV virus with some alterations for gene therapy. It's because we learned that this virus does a number of really spectacular and unusual things. So just learning how it works has payoffs. So I'm going to talk about two problems, two out of the many riddles which HIV is posing for us. The number of riddles is increasing with time, not decreasing. I'll talk about these two, the problem of the interrupted budding and the problem of the second shell. These are problems where there can be little doubt that physics is important. Not by itself, physics is important as an additional perspective which complements that of the life scientists. A physicist this would practically be impossible without the availability of something called PubMed, a database where you can study or read or enter thousands of medical journals. One price you pay is an enormous amount, a flood of information, which would, under normal conditions, be indigestible. So the role of the physicist here is very different from somebody who sits in a room, talks to experimentalists, and solves complex equations and makes prediction. It's more Sherlock Holmes. He's the patron saint for this kind of thing. You're provided with an immense number of clues. The vast majority of these clues are probably irrelevant, and some of them are probably wrong. And you're going to have to try to make sense out of it using a combination of guessing, testing. So without further postponement, let's start with the problem of the interrupted budding. So what you're seeing over here on the picture to the left is from 1992. I understand that I now have to push a little harder to the. This is a Mac computer, but my connector is not Mac, and I think that the company is letting me know its views about that. This is the birth of an HIV virus from an article of 1992. So you see here a line and a sort of a thick shell below it. Everything below this line is an infected host cell. And for HIV, That is normally a cell from your immune system, white blood cell. The only part from the virus you see over here is this thick layer, this sort of, it's actually a hemisphere. And you see these proteins, which are very large for proteins. Typically, you will not be, some of this stuff here is proteins, you won't be able, usually they're not that huge. So you see a thick layer of viral proteins. This line over here, which extends, which still covers this, these viral proteins, is called the cell membrane. And we're going to talk a lot about the cell membrane. Every cell in your body is surrounded by a membrane, and lots of compartments inside your cells are also very similar membranes. And HIV belongs to a group of viruses which are called enveloped membranes. That means that they are surrounded by a membrane which is basically similar to that of a host cell. One advantage is that it acts like a stealth device. Your immune system must try to find out the alien invader who came over the border. But why not carry a coat of the natives? So that's what HIV does. It has a coat which looks just like the membrane of a cell. This bar here is 1/5 of the wavelength of light. A fundamental principle of optics says that if something is this size, you're not going to see it under the microscope. That's a big bummer because we have an enormous amount of methods available for microscopy. We have to rely here on another sort of microscopy, which is based on electrons. They have a shorter wavelength, and so you can see it at a big price because you can't see it in a living system. These kind of sections are made by cooling it rapidly, and this cell is dead. So cryo-electron microscopy is the one way we can visualize HIV virus, but we can't visualize it in a living system. So this is the bud. We're talking about the problem of interrupted budding. So this is the start of the bud. If you wait for a while, this hemisphere grows, and begins to pinch off. That pinching off has just happened over here. Although you can see a lot of stuff in between. It's actually membrane, we'll talk about. This is called the immature virus. And the problem of the interrupted budding is that the virus can stay in this sort of state for a surprisingly long time. We'll call this the neck. It's like a neck. This is like the head looking at you. Here, 2 eyes. And the neck seems to be firmer than we would have expected. The HIV virus is unusual because it's a little bit alive. Once it leaves its infected mother cell, it changes, it grows, it matures. And this is the mature virus. And what you see here is something which HIV shows, and its family, its relations, but no other virus. There are 2 shells. There's an outer shell, which is responsible for mechanical protection, for docking, for all of the stuff where HIV is going to interact with other cells. But inside, you see a second shell. And the genetic material of HIV is in here. You also see that not all of these shells look similar. In fact, they're quite polymorphic. So there are 2 shells. So my second problem is, why ever do you need 2 shells? A respecting virus like common cold or flu, one is just fine. It provides the cell with mechanical protection. It provides, the virus. It also provides the virus with protection against enzymes which might chew up the genome molecules. By the way, I'm very used to questions. I know it should be at the end, but please ask them right away if I say something which is not clear to you. So please interrupt me. So these are the 2 problems I will discuss. This is a cartoon. Life scientists love cartoons. The books are beautiful with cartoons, but they are cartoons. So here you see the same process. This is the infected host cell. It's the membrane, curves around it. These are some proteins which are sticking in the outside, which are going to play a role later on. This is the budding. You see here this neck. Budding off has happened. By the way, this hole here is real. This is a virus which is raggedy. It has raggedy holes, and they also will be important. This is this step where it matures. It looks like this. You see the two shells, the outer shell with the membrane and some protein material, and an inner shell, and then this, that's the genome. So that's the mature infectious virus. There are two molecules in there of a type of genetic material called RNA. I'll explain that later. Here I only want to talk about this thick yellow line, the membrane. If you amplify this, magnify it, it looks sort of like this. It looks like it's a bilayer, so there's a layer of small molecules here, small molecules there, and these molecules are basically the same as the soap in your kitchen. These are molecules with one part which like to be in water. They're called hydrophilic. This is hydrophilic. And the tail groups like to be in oil. So this is kind of like a buttery, oily layer surrounded by these water-loving. They're called amphiphiles because they seem to want to go both ways. So this wants to be in water, that's an oil. And if you put this kind of molecules in water, then they assemble all by their little cells in these beautiful bilayers. So water is out here, and think of this as a very thin, it's about 100 angstroms, very thin layer of oil. It's very flexible, and there's a branch of physics which addresses this kind of structure. The kind of physics you learned in high school cannot deal with it. It's called soft matter physics. And I'm a soft matter physicist, and that's the perspective I'll talk about. So soft matter physics deals with these very deformable, soft things. Sure, as an expert on polymers, very soft, deformable linear polymers. These are very soft two-dimensional structures. The immature virus, here you see once again a cryo-electron microscopy image. Here it's enlarged, and you see now a little bit more of this internal structure. The membrane is out here. The RNA genome are these two red lines. There are two of them, and they're linked together, they're tied. The stuff in between is called the gag protein, and it's the hero or villain or whatever you like of the story. It is a very strange protein. Here it is. It contains multiple parts. These scissors here, we'll talk about it later, they're going to play an important role. I'm going to call this bit membrane associated because it binds to the membrane. This bit I'm going to call sticky because it holds the shell together. So these green bits, they are sticky, they stick to each other, and they make it into a kind of a capsule. This I'm going to call glue because it binds gag to the RNA, and so you can kind of see how the whole thing sort of holds together. It sticks to the membrane on the outside, it sticks to itself, and it sticks to the RNA. This forms completely spontaneously, by the way. There's no master who puts everything together, something mysterious, DNA coming here, and protein. You can do this in the lab, and it will work. Good. Enough about this is worth many lectures, and I'm not going to bore you with it. I only want to tell that this is kind of picture which you see in a biology book, but if you model it on the computer, and despite my stupidity with computers, this is what I do for a living these days, this molecule is not stable. It fluctuates in a crazy way. Here you see computer tracks. It sort of waves in the wind. The reason that happens is because we have thermal fluctuations. And it's a very important ingredient when you have these soft systems. You live in a very different world of thermal fluctuations, viscous loss. It's a different world. Everything is moving all the time. So if you model this on the computer, it doesn't look like that. It looks like this. And everybody wants to have more computer time so they can make longer trajectories for thermal motion. All right, so enough about gag. By the way, gag doesn't mean has anything to do with a physical reaction, but rather AG stands for antigen. Okay. Budding. So we're going to go back to budding and interrupted budding. And I was going to tell you there's a riddle there. But physicists don't think there, at least in my area, they don't think there is any riddle there. And I disagree. So why do my colleagues think there is no riddle? Because budding is very, very simple for lipid membranes. So we're going to do a little experiment. We take a lipid bilayer, and we're going to put two different kinds of lipids in there. two different kind of lipids. I'm going to call them alpha and beta, right? So we start with a perfectly flat bilayer, put in two sorts of lipids. Your bilayers have 35 kind of lipids, so I'm being very modest. So we just put in two, and let's see what happens. What happens is phase separation. It's like what you put water in oil, then you either get oil droplets in water or water droplets in oil or something more complex like salad oil. So phase separation. So only look at picture one here. Let's say there is much more alpha than beta. Then beta forms little two-dimensional drops, little plaques. And there is phase separation and there's a phase boundary. You might remember from your high school physics that there is tension along such a boundary. So you now have this interface tension, and what it wants to do is make it as small as possible. This interface tension has to do with the molecules over here are not happy. The alpha type molecules are not happy because they're in contact with beta, and the beta not happy because they're in contact with alpha. So this is a line of high energy which wants to be, it's like an elastic but wants to become smaller and smaller and smaller. And here is what happens. We call it escape into the third dimension. So you take this little two-dimensional puddle, you squeeze it, and pop, it pops up. There it goes. And from then on, it's just straight downhill. You form these buds, and they're going to come off. And this happens in your body a lot. So the buds pop out due to interface tension. Very basic physics, very simple. Budding is no problem. The final pinch-off, I'm overemphasizing it, is spontaneous. It happens all by itself. You don't need to do a thing. It just pinches off. To belabor the point, by the way, this is work by a German theorist, Reinhard Lipowski, in 1992. So it's been around for a long time. We think this is all correct. So we can do better by taking the concepts of soft matter physics and write down dynamical equations, starting here with a kind of toy cell. So I take a toy membrane in spherical form. I put in alpha and beta, blue and red. I'm going to put in more blue than red. And there's a set of transport equations I'm not going to show, which you can solve. You may have to do it on the computer, but this is basically what happens. So you start with a kind of fine mixture of the two types of molecules, and here you get phase separation, just like oil and water, pretty standard. You get lots of blue together and islands of red, phase separation. That's the first step. Now look at it, it's not quite round anymore. The reason it's not quite round, because here there are some really unhappy molecules. They are colored here yellow. They can be alpha, they can be beta. So this squeezing process has started. Here you start to see the beginning of a budding. Here it buds out, you get a hemisphere. Over there you see that already one of them is at the point of leaving. Here it's left. And this one is now close to leaving, just not so far after this one. This one is left. This one takes a little longer, but eventually it goes off. Then you have three little droplets, which are our toy viruses. And this is the final state. When it has reached this state, nothing more happens. It just sits there and you can twiddle your thumbs. So in summary, you get three successive budding events at this time step, this time step, and this time step. And after that, you just get relaxation to the final state, and it's going to stay there. So this was work done by colleagues from China, and this is the reference if you're interested. So this is exactly the Lipowski process. The tension causes these things to bud off. It's spontaneous, no problem. So, but there's a deep underlying principle here. That principle is the second law of thermodynamics. And that's what drives this thing. So the second law of thermodynamics states the following for these kind of spontaneous processes which you just saw. A system at fixed temperature, all biological processes of importance are at fixed temperature, at body temperature. That's what makes it very different. Everything is at one fixed temperature. So you need to do that at fixed temperature. So you know the second law has to do with entropy, but at fixed temperature, the key concept is free energy, which is a combination of energy and entropy. It's just the difference between the two. A system at fixed temperature can, but doesn't must, can evolve spontaneously if the process reduces the free energy. So here is it in graphical form. This shows the free energy, which is, as I said, combines the contribution of energy and entropy. Here you see it go down. This is the initial state. It has the highest free energy. This has a lower free energy, and this is the lowest free energy. So that's a spontaneous process. And this is a free energy minimum. It's going to stay there forever. Another word for this is perfect thermal equilibrium. And one of my colleagues, a famous colleague, said, if it is in perfect equilibrium, it's dead. That doesn't mean that it's alive here, but nothing more is happening once you're in a state of free energy minimum. So we're going to use this principle a number of times here. So with all of this, do we understand the physics of budding of HIV-1? And as I said, the consensus in my area is, yeah, it's okay. And here is what happens if you work with your colleagues in the life sciences. They bury you with facts, papers and papers and papers. And something comes quickly clear. There are some ugly facts, and here are the ugly facts. Unlike budding of lipid membranes, HIV budding stops. Roughly in this state. Here you see an electron microscopy image. It stalls roughly after the neck has formed. Yep. Didn't happen in the picture I just showed you. The final release, this pinch-off, involves recruitment of escort cell machinery that produces a contractile ring of proteins that's basically and pops off. Okay, you volunteer to read about the escort machinery, and whoa, I mean, it is protein after protein after protein, pathways. Wow, gewalt, as we would say. This is not nice. And perhaps this is not an area where physicists may want to work. Who needs all of this stuff? Then this, do you see this? Shouldn't be there. It's not there for lipid vesicles popping off. It has holes in there. Now, do you remember that I told you what drives it? It was tension, interfacial tension. Now, if interfacial tension is so important, it drives this whole process, then where do these holes come from? Because these holes, they should be closing under interfacial tension, and it doesn't do that. In actuality, the soft matter picture seems to be completely wrong. All right, good. Okay, you take a deep breath, and either you go to you go back to string theory, or you press on. We press on. So, is there some other thing? Well, when you talk to your colleagues, they say we believe that we know how it happens. Well, the way it happens is that these viral proteins, and there are other kind of proteins who do the same thing, they're kind of conical. They tend to bend the membrane a little bit. And it so happens that if they do that, they tend to move together. And we think we understand why. So if you embed conical proteins in a membrane, then the membrane begins to curve. So imagine that you have ice cream cones, and you put the tips of the ice cream cones together. It plays out. So if these are in a membrane, and they want to stick together, the membrane is going to curve. Now, the gag protein happens to be a sticky, remember the sticky bit? It's curvature inducing. If you have a couple of them together, it looks like an ice cream cone. And, I said here twice sticky, sorry about that. So, we need to redo the physics of budding. And it has to explain the phenomenology we talked about before. And gag is a veritable horror. It's a really bad guy. You don't want to have to think about all of this. So if there is a physical mechanism, you have to make it simpler. So you need a model. That's what physicists love to do. We make models. And we're going to make a toy model. I've always been told by people who evaluate me, never use the word toy model. People won't take you serious. Okay, hey, I'm tenured. Toy model. Okay. So here we have a toy model. The hell with it. And so let me start. To make a conical capsid protein, a conical viral protein, I just take 3 balls here, big, 3 smaller balls there, and green balls in the middle. And I'm going to call these hydrophilic, these hydrophobic, and these hydrophilic. Why do I do that? Because I want them to go into a membrane. And you remember the membrane is an oily layer. So this kind of structure wants to go into a membrane. Is it like HIV gag? Not really, but never mind. The next bit is the life scientists tell us that when they stick together, they splay out. So what you do is you make green attract green. And that sets a kind of cone angle, right? It sets an angle of curvature. So you can easily show that if you take lots of these, then the minimum free energy state, remember this concept, free energy? The minimum free energy of a bunch of cones is a sphere. And the radius of the sphere is determined by the angle of the cones. So the minimum free energy state is a kind of toy virus. Good. This is not my model. It was developed by Michael Hagen in Brandeis University, who did the simulations. I'm going to show you in a second. I also need the membrane, and I work in the same way. Here you see how a real lipid looks like, kind of vague, so forget about it. A lipid molecule, I'm going to make one thing which likes water, so it's blue, and one thing which is sort of yellow, like butter, which likes oil. And yellow attracts yellow, and blue repels blue, and blue repels yellow. And so let's call this hydrophilic. I apologize, you probably can't read it. Call that hydrophobic. You throw a lot of these in the computer, and you say, find for me the minimum free energy state. I'll tell you how that works, but for now just take my word for it that the minimum free energy, if you put 1 million of these and you allow them to float around like there, then eventually they settle down in this state. In the state I talked to you about before, where you have hydrophilic, and hydrophobic in between. So the bilayer state, we want that. Now the minimum energy state would be flat. But this is the minimum free energy state, which is seen kind of waving about. Do you remember I talked to you about thermal motion? This layer is waving, it's not flat. It's doing that to create, so to speak, entropy, and thereby find the real minimum of the free energy, because in the minimum of the free energy at finite temperature, this thing is waving around. So now I have the 2 components. I have the protein, I have the membrane, and now I start by embedding a lot of the toy proteins in the toy membrane at random locations. So I start here and I plunk here my protein. That's my model. And let's see what happens. In principle, I should be solving the soft matter transport equation. And as you say in the United States, forget about it. I mean, way too complicated. So what do? We are solving simply Newton's law. The Newton's law, which you remember from high school, for all the interacting particles. The proteins, which are made out of, I don't know, 12 balls, stick together. The lipids, which are made out of 3 balls. Just solve the equation of motion at a finite temperature, body temperature, say. We know how to do this on the computer. It's not pretty. I wish I could do it analytically, but I can't. And this type of computation is called molecular dynamics. So I'm going to show you, hopefully, how this works. So there you see it. And I'm going to stop this. And I watch this go. So look carefully. Here you see these brown spheres. They are membrane proteins who haven't yet joined the growing shell. I'm not showing them here. Here you see simply this shell growing. These particles are diffusing randomly away. These bees, which you see over here, are just lipid molecules which got out because of thermal motion. You see the neck forming. Do you see how quickly it went? And remember, has anybody read the story by Sherlock Holmes, the dog that didn't bark? Here, the story is nothing happened for a long time. Did you see the flash? That meant that you can sit, you sit there and the computer works away for an hour, nothing happens. Then suddenly, something happens. And the virus appears. And now I need to stop this one. And I'm going to show you the other one, which shows you cross-section. And it'll tell us a lot about what happened in this strange process. So here you see the free proteins. They're trying to get in there. They're just randomly moving around. Some of them succeed in joining the club. This is a tight club, considered like an elite or something like that, or in a high school clique. They want to join it, and they're diffusing around. The membrane is evolving and becomes more and more of this kind of shape. And it kind of begins to seem that it's harder and harder for these outsiders to become part of the inside club. They try, every now and then one succeeds, and this is the key event. I should have stopped it at this point, excuse me. Too late. Did you see what happened? What happened was that you had this neck, and suddenly the two sides of the neck, by a thermal fluctuation, came together, and whack, off it went. And here you can see that the virus has formed. Do you see the hole there, by the way, where it should be? And it will go off in a second. As far as I can tell, this little simple model does all the things which you see in HIV, without having to worry about all of the complexity. The simple model seems to reproduce the phenomenology of HIV, including the ragged holes. Depends on this parameter. Remember, there was a parameter, how strong you couple them together. If you make that parameter stronger, you see lots and lots of ragged holes. The free energy picture. When I show this to my colleagues, they say, oh, we know this, come on. It's a metastable state. You know, your neck is metastable, it sits there for a long time, until there's a thermal fluctuation, brings you over, and we are happy. There. That's kind of 19th century physics. Is the next state metastable? We'll show you that it isn't. There's new physics here. Good, I'll try to convince you of that. But we have to make a detour past some mathematics. Carl Friedrich Gauss was greatest, or people argue about it, but maybe the greatest mathematician of the 19th century. And I'm going to use a little bit of mathematics from him to analyze these pictures we got from the molecular dynamics. So look at these three, a sphere here, a cylinder there, and this looks like a cooling tower of a nuclear reactor. You may have heard of nuclear reactors in your history books. So I'm going to take this point, and you can see there's a circle there. I can draw the circle just at this point. I'm going to call that radius R1. I can also draw a circle perpendicular to that, and it has a radius R2. And Gauss defined the concept of Gauss curvature, which is simply 1 over R1 times R2. The minus you put there if the center of this circle and the center of that circle, if one is inside and one is outside. If both are inside, you give it a positive sign. So this structure has a negative Gauss curvature, and a fancy word for it, a hyperbolic or saddle surface. This structure, suppose you can go from this structure by making this radius larger and larger and larger until it is perfectly straight. So this is kind of a degenerate case of the hyperbolic surface with R2 infinite, which means that the Gauss curvature is 0 because you make R2 extremely large, and that's 1 over a large number. Finally, the circle, you can take two directions here, perpendicular to each other, but all of these curvature radii are the same, so the curvature here is 1 over r squared. So this is a positive Gauss curvature, this is 0 Gauss curvature, and this is negative Gauss curvature. What does it have possibly to do with HIV? Sorry. The most important bit is there's a theorem. The theorem by Gauss says that if you have a closed surface, like a membrane, closed membrane, then the average of the Gauss curvature over the surface is 4 pi divided by the area. The professionals will not immediately recognize it, but it's the same thing. This is only true for a closed shell with no holes. You might worry about the holes I talked to you about. I'm going to apply this theorem to the lipid membranes. Okay, so here's the picture I saw from the simulation. I'm asking Carl Friedrich, what would you say about this? And Carl Friedrich would look at it and said, the first thing I would say is that these proteins really like regions with positive Gauss curvature, spherical regions. Why? Because they're clustering. They're coming there, they're minimizing their own local free energy per protein. They're very happy over here. No surprise, because by construction, a state of minimum free energy is the sphere. So they're very happy over here. These are happy campers. Next, you remember that I emphasized that once the neck formed, they don't seem to want to be here. Okay, so proteins hate membranes with large negative K. And finally, we have here a flat membrane, which is sort of like a cylinder, so K equals 0. Proteins are sort of okay, like the three little bears, middle one. So proteins are sort of okay, not really happy. They would love to be here, and they just seem to be unable to. Now, this makes a lot of sense because you design membrane proteins to get that state of minimum free energy. A membrane protein likes to have positive Gauss curvature. So it is perfectly reasonable that they don't want to go to places with negative Gauss curvature. Individual proteins need to overcome a geometrical energy barrier over here before they can join the bud. I call this molecular hazing. Imagine a fraternity. This is the fraternity, guys. The first years, the plebes, I think they're called, they're going to get hazed. They have to go through this. They want to join the club, but they're going to have to deal with some issues. They have to get over here. This barrier becomes narrower and narrower as the last few get through. So this barrier gets bigger and bigger. The free energy per protein is apparently very high in this negative region here, very low there, and kind of intermediate there. Growth stalls because nobody except a super protein can make the jump anymore. Okay. So, when I give this talk, people, how about evolution? How about evolution? This fantastic idea of Darwin. It can do anything. Could a budding virus not avoid having this negative Gauss curvature region by some sort of molecular evolution, some, you know, who knows what? Okay. No. Remember Gauss-Bonnet. The average Gauss curvature over the whole membrane, I mean this whole closed cell, is 4 pi divided by the membrane area. The number of lipids is fixed, so that means that this is a fixed quantity, which means that the average K is a fixed quantity. So, if you want to have a bud appear, let alone a virus, then you need to pay a price. Even though this is very nice for the proteins, by the Gauss-Bonnet theorem, you generate unavoidably a region of negative, because the average has to stay the same. If you put something positive there, something negative has to be the same, has to cancel it. So there is no way you can avoid this region of negative Gauss curvature. Gauss will say, so how shall I say it? Evolution cannot make pigs fly or elephants fly. It cannot violate fundamental principles. So evolution cannot help taking care of that. So in conclusion of this problem of interrupted budding, the interruption of the budding of enveloped virus is an intrinsic feature of budding, driven by the mechanism of curvature-inducing proteins, plus a theorem by Gauss. Evolution. Darwin is not able to beat Gauss, but evolution finds a jury-rigged workaround. It borrows, steals, hijacks a cell machinery. Remember this red line pinching it off? That's a kind of thing which happens in the cell, and there is this dedicated escort machinery, which is very complex, and you need it to get over this energy barrier, to send off this virus into outer space. So that suggests a solution to HIV. Should we not suppress escort? Okay. Unfortunately, escort machinery is essential for normal cellular processes called endocytosis. It's the same physics as that of the budding of enveloped virus. So you see how diabolically clever, if you can call it clever, how diabolically it borrows machinery which you cannot suppress. If you would suppress the escort machinery, or if you had mutation which turns this down, so you had less chance of AIDS, then you pay a price that normal cell processes, which involves this budding, are also stopped. So even though Darwin cannot beat Gauss, he completely negated the negative Gauss curvature problem by using available machinery which you cannot possibly shut off. So in the end, Darwin does beat Gauss. But in this trip, we have learned that there's an underlying physical principle which makes life quite hard for HIV, and it may suggest other ways to inhibit HIV. I cannot think anyone except for this. Maybe you. We're going to go on. Is there a question?
Why don't the purple molecules, why do they dislike the negative curvature?
I deduce that from this picture. Because in the Boltzmann probability distribution, particles will go to a place where they have a low energy or a low chemical potential. From the fact that the particles don't come here, it means that the chemical potential must be high there. So,
do they repel each other at close range?
What?
Do the purple proteins repel each other?
No, they attract each other. So think about it like this. These purple proteins like to make a spherical shell. Suppose that you have a few of them over there. Then in one direction they like it, but in the other direction they completely curve in the wrong way because they have to form a saddle. And so they're trying to close the saddle, so they are very unhappy in this negative Gauss curvature regime. The whole point of a membrane protein is to create a spherical vesicle. So unavoidably, it has a low free energy if the Gauss curvature is positive. Unavoidably then, they have a high free energy if the Gauss curvature is negative. So you cannot design a protein which generates curvature, spherical curvature, at the same time while this protein also might like a saddle surface. It's geometrically and physically impossible. You're at a dilemma. If you design your proteins to make a spherical structure, then unavoidably these same proteins will going to avoid a region with negative Gauss curvature. Am I okay? Good. Any more questions about this before we move on? Please. Okay, we're going to move on. The problem of the second shell. So here you see, once again, a cryo-EM picture, more advanced. Here you see the same picture exposed in 3 different ways. We call this tomography. You might have heard of tomograms, which you may undergo it yourself if you get scans. In tomography, you take different projections and then you combine it to construct a 3-dimensional realization. I'm horribly simplifying. And the question is now, why do you have 2 shells? So you have this blue spherical shell, which is this membrane with some membrane proteins, and then there's a second shell inside here. These are macromolecules which come along for the ride. Some of it just stolen from the host, some of it just random, proteins from the host. So what is the point? I mentioned before that decent RNA viruses, they have 1 shell. So why do you need 2 shells? Okay. First, you interact with your colleagues in the life sciences, and they have done lots of beautiful work. They reconstructed. Notice these are different second shells, and you see that they're not all of the same shape. They come in all sorts of shape. They come in a variety of cone-like shapes. That by itself is a very interesting story, but not what I want here. Importantly, they don't look like the right kind of shell. They're permeable. Do you see these holes? They're permeable for small molecules. In a decent shell, it allows water to go through, maybe salt, but that's it. You want to protect your precious genome from horrible enzymes which want to cut it up. You remember the scissors? Those really exist. They're scissor-like genomes, scissor-like enzymes. You call them RNases. You need to keep them out. So to make this thing permeable looks weird. We're going to go on to the next clue. Remember, we are Sherlock Holmes, we are collecting clues. Of course, I'm only telling you the clues which are useful. The other clues, you just argue with your friends until you're blue in the face. Here, the second clue is actually, it doesn't look obvious. Here you have a second shell, and there's something dark in here. That dark stuff is the RNA. Those are 2 RNA molecules, For the professionals, that corresponds to here to 20,000 RNA nucleotides. You may remember the code of Crick and Watson. You need this code is expressed in terms of nucleotides. So this tells you how much information is stored on the genome molecule. It's like a manual to assemble it. But here we're going to talk about sort of the material science of HIV and the consistency of this stuff. It is blobby. It is jello. It doesn't have any shape or stuff. It's like gel. It's a blob of RNA genome molecules, and it can adopt any shape. It can fit in any of these things perfectly happy. It's like soft stuff. Okay. Please keep that in mind. RNA is soft. Back to high school. I have to go through this. This is called the central dogma of genetics. So let's walk through it. All your genetic code is stored on DNA. It's your library. It can replicate. There are special enzymes which do it. If you need to make a kind of a certain protein, what happens is that you make a copy of a certain section, which is a gene. That copy is in the material form of an RNA molecule. This is double-stranded. You may remember the double-stranded, the double helix. This is single-stranded. So this is just a single strand of nucleotides. This RNA molecule, which is much shorter than the DNA, because the DNA may have 30,000 genes, and this is just one gene, it's translated. Another kind of enzyme turns this bit of information into an actual protein. It's basically like a player piano. You put in instructions, out comes a protein. And that is the central dogma first developed by Crick and Watson in their shattering paper of 1956, I think. Good. So here we are. This is the basis. This is what every student in molecular biology gets to see first. Now we turn to HIV. Okay. HIV has a very special enzyme. It's called reverse transcriptase. And it does something which should be impossible. It can take single-stranded RNA like this and turn it back into DNA. And this special enzyme is actually inside our second shell. It is packaged with the second shell. It's quite big. It sits over here. This is roughly according to size. So what it does is it takes this viral genome, and I told you that HIV has an RNA single-stranded viral genome, and then turns it into a double-stranded DNA by this process of reverse transcription. Reverse transcription was first proposed in 1975 by Baltimore and Temin. It took years before people really accepted this violation of the central dogma. They got the Nobel Prize. In fact, I'll show you later that the first indications of this process date from 1944. Wait for that. Why would HIV do something like this? It is so insidious, so devilish, that I'm going to wait for you before I explain it, why it does turn RNA into DNA. It is really evil. Okay, so wait for it. For now, we're just going to see what the consequences are of turning RNA into DNA for the second shell. Okay, for that I have to tell you just a little bit about the materials property of RNA versus DNA, particularly single-stranded and double-stranded. Remember, single-stranded RNA was blobby jelly. It's flexible and it's compact, and we understand exactly why. Here is how it looks like, this strange spidery structure. This is a viral, not HIV, it's another viral, as a single-stranded RNA molecule. It's branched, which makes it compact, it shrinks it, and it's bubbly, which makes it very flexible because along all of these single-stranded bubbles, you can fold it as much as you like. The reason for that is you have to look at the molecular level where you have the genetic code, which for RNA is A, U, C, and G. G pairs with C, it forms a hydrogen bond, and A pairs with U. So if you have a single strand of RNA and you let it find its free energy minimum, it can do that in 1 trillion different ways by finding the best possible match. So everywhere where you see this little thing sticking out here, this is actually like this. So they're magnificently complex, but compact. That's all we really need to know. DNA, double-stranded DNA, is extended. It's rigid, no branches, and few bubbles, particularly at low temperatures. And the double-stranded DNA molecule is much larger than this blobby RNA molecule. Here you see a viral DNA molecule plated on a surface and displayed by an atomic force microscope at the scale of the second shell, roughly. And you can see there's a problem. We need to squeeze all of this in there. That doesn't look good. In fact, it's impossible. You cannot take a double-stranded viral RNA and squeeze it in here. It would be totally impossible. You need to condense it, like condensed milk. And you do that with glue. If I use a common language, I'm a Dutch farmer, so we have glue, we take DNA, we put in the glue, and what you see is something magical. The DNA rolls itself up, like a garden hose. It's just like a sticky garden hose, and it rolls itself up and becomes much more compact. You can take a cross-section of this, and you see this beautiful hexagonal pattern. So this is a cross-section of this sort. It's a torus, and here's the key point. It's rigid. It's a hexagonal, it's a crystal, it really is. By the way, inevitably, when you try to wind this around, you run into some problems. There will be, this crystal is actually defected. But never mind that, that's fun. So, if it was condensed, then the DNA might fit in the second shell. But where would the glue come from? By the way, the glue molecules shown here before, they are called spermine, and the males of this audience have a lot of spermine around because that's how you condense male sperm. So these male sperm cells, they have a lot of wound up DNA, and it's in these little heads you may have seen with the little tails from the Woody Allen movie. So spermine is the molecule which is used, and the key point is these are little molecules and they're positively charged. By the way, it was discovered by my countryman Anthony van Leeuwenhoek. When he reported some of these things, the Royal Society in London said, he's a drunk sot. So, I had to get it off my chest. Here's how it works. So you have this DNA. DNA is heavily negatively charged and it repels. But if you put these positively charged spermines in between, you can make a beautiful crystal. You just charge neutralize it, plus, minus, a lot like salt. So you can condense DNA with spermine. And here's the problem. Where does it come from for HIV? And here is the trick. Remember this picture, these scissors? Okay. This little bit, it's called the nucleocapsid sequence. I called it glue before, has a charge of 3.5. When you make the capsid first, this immature virus, you have about 4,000 copies of GAG. When you cut that, you have 4,000 of these molecules, which is enough to condense the DNA. Isn't it beautiful? So, even though you churn out DNA from the RNA, and it would like to explode out of the shell, you provide it at the same time with glue. So, now comes the key question. Does the DNA, condensed DNA torus, by the way, these tori are always roughly the same size, does it fit in the second shell? I've drawn it as best as I could to proportion. Here's how the viral genome looks like when it's condensed, and here's the second shell. It just about doesn't fit. It's a little too big. The diameter of the double-stranded DNA torus is somewhat larger than the maximum diameter of the second shell. That can't be a coincidence. Okay. So, if you just go on, and remember this is rigid, this is not like the soft, squishy, blobby RNA, it's a rigid crystal. It's like diamond. It grows, it grows, it grows. And if it reaches this size, if you're turning RNA into DNA, it's going to push these edges out. So, could reverse transcription cause the second shell to burst? It would be really weird, right? Why would, it's like suicide, viral suicide. That sounds too weird. But for once, there was somebody who was willing to do the experiment. Itai Rousseau in the Weizmann Institute did the experiment. So he has the second, not because I suggested, by the way, he did it all by himself, he had read the note. You take now a second shell, the RNA is in here, and it turns out that you can turn on reverse transcription. So here is the second shell, all single-stranded RNA, this is the same scale as before, and you can poke it with an atomic force microscope and it's pretty rigid. Now you turn on transcription. The characteristic time scale is about half a day, 15 hours. And after about 15 hours, suddenly it bursts, boink! And the way you know it is because when you poke it, suddenly it becomes very soft and squishy, rather from stiff. That's because it has, the DNA has transcribed, has burst, and now it has lost all its tension, its pressure, and it moves out. These are repeats for another cell and for another cell. This one after 15 hours again burst. This one after 15 hours burst in two different places with the DNA coming out. So something, this bursting has something to do with the function of the second shell. By the way, you should ask the question, how is this possible? How can bursting a shell lower the free energy? It doesn't make any sense, right? This is a perfectly spontaneous process. When Itai turns it on, it evolves and you end up with this broken mess. So that doesn't make any sense. Does it not violate the second law? Here you see reverse transcriptase, this enzyme, in its full horrific glory, his diabolical majesty. RNA and DNA, and here is the point. Reverse transcriptase is not just any old enzyme which promotes a chemical reaction, it's an engine. It's a very efficient engine which can convert chemical energy obtained from the chemical reaction which forms the DNA and transform that into mechanical force. So the bursting of the virus is due to the reverse transcriptase, which takes chemical energy from the environment, just like the engine in your car, takes small molecules and takes their energy away, and the total free energy is still reduced. So you see how HIV keeps throwing surprise after surprise after surprise at you. We're nearly done. Let's go back to the life. What's the point of this whole stuff? And I think I know. So here you see the complete life cycle of HIV. We've already gone over this part. That was the mature virus. Here you see it. Zoop, there it's back. It recognizes another cell. This is not the same cell, of course. Another white blood cell. It enters by fusing its membrane with that of the cell. Very nice. And there we have the second shell all by its little self with its RNA inside. And according to the textbook story, it magically falls apart. The genome, RNA genome comes out. Reverse transcription kicks in and makes it into DNA. So now I can finally tell you the diabolical cleverness of HIV. This is double-stranded DNA, which is just like your genome. So what happens is that this double-stranded DNA, which comes from the virus, gets integrated, inserted in here. It is mutating the cell. So these white blood cells are mutants. Eventually, this gets expressed and translated, and the cycle starts again. So it alters the genetic code of the cell. You may or may not like to know that your own genome has lots of this stuff in there. What has happened is that some of these viruses decided we don't need all of this stuff. We just ride along with the host. Okay? We don't need it. We just let you do the reproduction, which might be fun or not, and we just go along for the ride. So we reproduce ourselves. The word is called selfish genes. So this was discovered, by the way, by Barbara McClintock. I'll tell you more about her. Here, though, I only want to notice that the uncoating precedes reverse transcription. It's wrong. This textbook story was shown by Itai to be wrong. It's the other way around. Reverse transcription causes the uncoating. And that's the key to the story. I still want to show you Barbara McClintock's picture. This was done in 1944. She was studying mice, and she discussed that the genetic code, or the genome of some mice that's corn, excuse me, of some corn was not stable. It seemed to move around and jump around, and she called them transposons. And the males ignored her, of course, for decades. And here is the explanation. The mice had these, had other, not HIV, had other retroviruses, and it had these inserted viral genomes, including the replicate reverse transcription. So that's encoded in here. So what would happen is RNA would come out, reverse transcription would make DNA, would go back in, and you have the gene in a different place. So the gene jumps around along the genome. So that's called a transposon, and that's what you have. So you carry reverse transcription with you, for sure. It's a significant part of your own genome. Okay, so end of the story. Conclusion of the problem of the second shell. Disassembly of the second shell is driven by reverse transcription, and it's the motor protein. That's nice. No question anymore about it. What is the function of the second shell? Here's what we think. It is a reactor vessel. It is not at all there for mechanical or chemical protection. Well, at least not mechanical protection. It's a reactor vessel where you have a high concentration of reactants. The key is reverse transcriptase. Reverse transcriptase cannot escape. So you have here the RNA, here we have reverse transcriptase, and it cannot diffuse away, which would be disastrous. So your precious reverse transcriptase is kept in your reactor vessel. These are holes, and through these holes, DNA nucleotides can come in. That's the feedstock of the chemical reaction. It protects the viral RNA from enzymatic digestion. Inside your cells, you have RNases which chop up RNA. They can't get through. The holes are too small for that. It fractures when reverse transcription is roughly complete. So it kind of measures, it gauges. And when it's done, it bursts. Can we test this picture? Yes, it turns out to be possible. And of course, it has also to do with we want to inhibit HIV. So we would like to fool around with this vulnerability of this weird second shell. So there's two ways you can do that. One is change the stiffness of the second shell. You do that by mutation. So you can make the sticky bit of gag a little different. You can make the shell either stiffer or less stiff. So this is an experiment which was done. Here you see HIV entering the cell. If you put in a mutant which makes the second shell less stable, then it uncoats deep inside the cell where all of these RNases are around and they all cut it up. Nothing happens. You can also make a mutation which makes the cell hyperstable. It doesn't uncoat. It's like a chick which cannot get out of the egg. The shell is too hard. Doesn't work. You can get the same thing by inhibiting reverse transcriptase. There are chemicals which can stop reverse transcriptase. These are actually anti-HIV drugs, and they have the same effect. Because you're not exerting this pressure, it just stays there. Productive infection actually requires that this second shell docks onto the nuclear membrane and then releases its DNA for insertion in the host cell. You would have guessed that if we did this experiment and with all this success, why are we not writing this letter in Nature? That's because the experiment was done in 2009. Okay, so it's already known. It's a little bit of a disappointment, but it doesn't matter. It confirms what we knew. This was one of my colleagues. I'll tell you about it in a second. She found this article, which seems to completely nail the picture which I told you about. So these are just two of the riddles, and we've come to the end of the talk. I mentioned to you the enormous amount of information. It's impossible for a physicist to work in this area without guidance. Julia Rosina is a physicist who works in the Ohio State University in an HIV lab, and she has an uncanny ability to go through all of this literature, which would just kill me, and tell us what is important, what you need to read. So without having such a person there, you wouldn't get anywhere. I want to thank my group. Sanjay Dharmavaram, who's now in Bucknell University, who did a lot of the hard work. Celine Bao Shen, who's in Stanford. my colleague Joseph Rudnick, and my graduate student Chen Lin from the chemistry department. And with that, I want to thank you. I'm afraid I went over time, right?